Okay, thank you so much for the introduction, Lisa, and thanks to everyone for coming out tonight. I'm so excited to talk about malaria and evolution with this audience. So I'm going to talk about how evolution can help us understand malaria as a disease. It's one of the diseases with the richest evolutionary history, I think, to think about. And also how we can use evolution as a perspective or a lens to refine and improve the ways we control the disease. Before I talk about those things, I just want to mention a little bit about my background and how I got into malaria and computational biology. When I first came to Boston, when I started graduate school, I actually was working on pufferfish. I was interested in why pufferfish have the smallest genome sizes of any vertebrate. And when I finished my thesis, I wasn't sure that I wanted to work on pufferfish for my entire career. But they're lovely fish, but they don't have a major public health impact, sushi preparation accidents aside. And I was amazed and pleasantly surprised to find that the skills that I'd learned to study pufferfish genomes, of analyzing genetic variation in populations, comparing genome sequences to each other, had ready application to public health. So studying infectious diseases. When I first moved to the Broad a little over 10 years ago, I worked on pathogenic fungi that are illustrated here in the middle of this slide, Cryptococcus and Coccidioides. But I quickly moved into malaria, studying the Plasmodium malaria parasite that actually causes the disease, and the Anopheles mosquito vectors that transmit malaria from person to person. And the reason why I was able to move between these different fields without completely retooling myself, these different disease systems, was because all of these study organisms I investigated by a common set of tools, a common set of population genetic tools for measuring and understanding variation and applying that to phenotypes, traits of an organism. In pufferfish, that was small genome size. In malaria, that could be resistance to drugs. So there's a lot of transitivity of that population genetic tool set. Because I'm a computational biologist, I don't have a laboratory. My laboratory is my laptop computer, which is right here on this desk, and the Broad computing cluster. And I therefore rely on a lot of collaborations. So I just wanted to start this talk with an acknowledgement of all the collaborators, the wonderful collaborators that I have in the Broad and Harvard and Boston community and at other institutions and universities around the world. So why malaria? Malaria is a disease that most of the people in this room probably don't think about very often, but which is a fact of daily life for almost half the world's population. The WHO statistic is that almost half the world's population is exposed to malaria in a given year. So the most recent statistics are that malaria causes about 600,000 deaths per year. Most of those are children under the age of 5 in sub-Saharan Africa. And apart from that mortality, it causes a lot of morbidity or sickness. So there are 200 to 300 million infections per year. And these are more serious than just a head cold. All of this illness imposes a significant economic burden on countries where malaria is endemic. And Jeffrey Sachs estimated that there's a significant impact of a high burden of malaria on GDP, with perhaps up to a 5-fold reduction in GDP. So malaria impedes the development of countries, and tragically, it's development that can actually help control malaria through reduction of mosquito breeding sites and improved infrastructure like window screens and such. And finally, because of our long association with malaria, malaria has had an impact on human evolution. So even if in this room, almost no one has ever seen a person infected with malaria, and certainly has likely not been infected with malaria. I'm not saying that applies to everybody, but it applies to me. All of us in our DNA surely bear some kind of mark from evolution's interaction with malaria as a selective pressure. So the symptoms of malaria are varied. There's a constellation of symptoms, but the signature symptom is fever. And malaria used to be referred to as tertian or quartian fever, denoting the periodicity of the number of days in between spikes in fever and chills. And that was the difference between tertian and quaternary malaria was related to the two different species of parasite that have different cell cycle lengths. So fever is most significant, but there are many other symptoms that are debilitating from malaria infection. And these are captured in this painting from the French artist Ernest Hébert, who spent much time not in Africa but in Rome. And it's not often recognized that malaria was formerly a disease not just of tropical areas but subtropical areas and temperate parts of the world. And in fact, the southern half of Italy was extremely malarious during the summer periods. Malaria has killed many a pope. And this is a painting of country people who lived in what were called the Pontine Marshes in the periphery of Rome, and which, until they were drained by Mussolini in the 30s, were a source of significant malaria infection every summer. And you can see this in the kind of forlorn, melancholy expressions on the people in this painting. So marshes, wetlands have long been recognized as being associated with malaria. But before we understood that malaria was caused by an intracellular protozoan parasite, and before we understood that it was caused by mosquitoes, it was thought that malaria was caused by the bad air, by the miasma that came out of wet areas. So it wasn't actually until the turn of the 20th century that work by Ronald Ross, a physician in the Royal Navy, and Battista Grassi, a zoologist from Italy, uncovered the fact that it was mosquitoes, and specifically, not all mosquitoes, but just mosquitoes from the genus Anopheles that were responsible for spreading malaria from person to person. So this was a key discovery. Ronald Ross actually got the second ever Nobel Prize in Medicine to be awarded in 1902 for this discovery. And this discovery that mosquitoes were the vector of malaria has led to a lot of successful control of malaria and elimination of malaria from many parts of the world. So, so mosquitoes are what transmit malaria from one person to another person. But malaria has a fairly complex life cycle within a single host. So after a mosquito bites a person and this malaria parasite sporozoites migrate through the bloodstream, they first lodge in the liver. They invade a hepatocyte cell, a liver cell, and they undergo a few stages of replication in different liver cells before they emerge and become merozoites, or blood stage infecting parasites. And it's this blood stage infection where they live inside red blood cells, the red blood cells that carry oxygen and CO2 around your body, that causes most of the symptoms of malaria. Because they can undergo this cycle in red cells of invading, replicating, and then bursting those cells, and then all the progeny then go reinvade other red cells, they can build their population up to an enormous level within your body and cause anemia and a host of all the other symptoms that I previously mentioned. Some small fraction of those red cell infecting parasites differentiate into sexual gametocytes, and it's these differentiated sexual gametocytes that are imbibed by a mosquito that bites an infected person, and they undergo another part of their life cycle inside the mosquito to be reinjected as sporozoites into the next person. So it's a lot of complicated biology. It's not surprising that it's only been about 100 years that as a species we've kind of understood the nature of this fairly complicated disease. So as I mentioned before, malaria is an old disease. So this is a nice way of understanding the stages of disease acquisition. This is a figure from Nathan Wolfe and colleagues from Nature from a few years ago. We don't commonly think of diseases as phenomena that come and go in terms of afflicting us as a species, but they can be transient. We don't always have the same diseases, and many of the diseases that we have now as a species in various parts of the world have been relatively recently acquired. So diseases pass through stages of only being acquired from animals, like rabies. You never get rabies from the person sitting next to you in an auditorium. You get it from a dog or a bat or some animal that's infected. to Ebola, where there's perhaps an outbreak that's sparked by an animal-to-human transmission event, but then human-to-human transmission, to diseases like HIV, which might have originally been acquired from chimpanzees or other primates, but now are exclusively spread from human to human. So malaria is very firmly in this stage 5 of being an exclusive human agent, and it has been in that state for a very long time. So through some recently published genetic work from Beatrice Hahn studying the malaria parasites of great apes in Africa, we now know that Falciparum malaria, the species of malaria that causes the most mortality, is one that we acquired from gorillas. There's evidence that perhaps a single interspecies transmission event, mediated no doubt by a mosquito, which was likely not even noticed by the very first person to be infected by Falciparum malaria, happened sometime in the relatively recent past, maybe tens to hundreds of thousands of years ago. That's relatively recent in evolutionary terms, but we know that over the course of hominid evolution in Africa, even as a pre-human species, we were likely infected by multiple species of malaria at any given time. But this species of malaria that in contemporary times causes the most mortality and which is the biggest target for malaria control efforts, we've had for perhaps tens to hundreds of thousands of years. So that's long enough for evolution to happen between us and Falciparum malaria parasites. So Ebola and HIV are relatively recent diseases by comparison. The human population has not really evolved to a significant extent around either of those relatively recent diseases. But we have evolved a host of adaptations to our longer history with malaria. And malaria parasites reciprocally have evolved to specialize in their parasitization of humans. Part of the reason why malaria is such a bad disease, especially in Africa, is that's where there are the most efficient vectors of malaria. In Africa, there are mosquitoes, most notably Anopheles gambiae, that are highly specialized to live and breed in human-created habitats, and who love to bite humans much more than any other animal. And those make the most effective vectors of malaria, and that's partly why disease intensity is higher in Africa. So it's that adaptation between mosquitoes and us, and between parasites and us, and moving around this triangle that causes this disease. And this is what makes evolution an important perspective to consider when thinking about how to control malaria. So the idea that infectious disease could actually impact the evolution of a species was first promoted by Charles Darwin. This was an idea that was found in various places in The Origin of Species that was published in 1859. That's a big, thick book, so you can be forgiven if you read that and you didn't exactly notice this specific passage. But it was another evolutionary biologist, J.B.S. Haldane, who in the 1940s noticed that the distribution of certain maladies, like thalassemia or sickle cell disease, the geographic distribution of certain maladies was eerily similar to the geographic distribution of high malaria transmission intensity. And you can see in this top map is a map of the temperature suitability for Plasmodium falciparum malaria transmission. And in the bottom map is a plot of G6PD deficiency. This is a type of anemia that is related to malaria resistance. Okay? So this was the so-called malaria hypothesis, and not too long afterwards, in 1954, the biologist Anthony Allison demonstrated that, in fact, sickle cell disease did confer protection against malaria. So I'm going to go through some of these examples of how, as a species, we have evolved in response to this terrible disease, this disease that incurs high mortality in children and which we've had an association with for at least thousands of years. These are the perfect ingredients to cause an evolutionary response. So I just mentioned sickle cell. This is probably the malaria resistance adaptation that most people are familiar with. If you have one copy of the sickle allele, which causes a mutation in the gene encoding hemoglobin, in the hemoglobin protein, if you have one regular copy, one healthy copy, and one modified sickle copy, you are resistant to malaria. If you have two modified sickle copies, you have sickle cell disease. And this is manifested as these funny shaped little sickle-shaped red cells, which lead to to various symptoms. I think I pushed a button here. All right. There we go. OK. If you ever push the wrong button, just push it again. So in addition to sickle cell, in addition to modifying the proteins that compose hemoglobin, hemoglobin is the molecule that is pretty important to all of our existence. It transports oxygen and CO2. In addition to actually changing the structure of that protein to incur resistance, various human populations have also acquired mutations that lead to a deficiency in hemoglobin. So the protein sequence and structure is perfectly fine, but the level at which those genes are active has been turned down. And so this causes various thalassemias, known as alpha thalassemia, beta thalassemia, gamma thalassemia, because the mutations that lead to a decreased production of these genes have arisen many times in different parts of the world. When the malaria parasite invades a red blood cell, it eats the hemoglobin. All right? This leaves a lot of very reactive molecules in the cell. And one way that we can protect ourselves against malaria is by decreasing the amount of this enzyme called G6PD. And by decreasing this enzyme, it creates a hostile environment for malaria parasites inside the red blood cells once they start to attack that hemoglobin. So this causes anemia in humans that carry this allele, but it also confers protection against long-haul infection. And then finally, blood groups. So just in the last 10 years, there's been some evidence to confirm a long-held suspicion that certain blood groups confer resistance to malaria infection. And anybody in this audience who has the O blood group should know that that allele, that variation in the human population may have arisen originally to reduce malaria infection or the intensity of malarial disease because it prevents red blood cells from carrying certain antigens that the malaria parasites use to cause infected red blood cells to stick together and sequester in various parts of your body. So there's a host of polymorphisms, of variations in human populations, in red blood cells in particular, that we can trace to evolutionary pressure for resistance to malaria infection. And one of the more dramatic ones, which is still a bit, I think, contentious because it's an evolutionary story and we don't have a direct way of testing this, but there's this long-standing hypothesis that the dark gene, the Duffy antigen receptor, is actually turned off. So people of African descent have this gene, but they also typically, in most cases, have a mutation that prevents it from being expressed in red blood cells. This is important because it's this dark protein that lodges on the surface of red blood cells that the Plasmodium vivax malaria parasite uses to bind to when it's trying to sneak its way into the red blood cell. So likely to prevent malaria infection, this dark protein has just been totally shut off. And as a result, vivax malaria is virtually absent from sub-Saharan Africa. This is a case where, at least against one species of malaria in one part of the world, humans have evolved a very significant, effective level of protection, if in case this was the agent of this dark expression mutation. So it's not just one way. Malaria parasites have not only been influencing our evolution, we have been influencing the malaria parasites' evolution, and especially in the last century, as we've been developing drugs in order to control malaria and treat ourselves. So all of the major drugs that have seen widespread deployment to treat malaria or to prevent malaria have triggered evolution in malaria parasites, evolution for resistance. So chloroquine, which was a wonder drug in the 1950s and which was used in the first major malaria eradication campaign conducted by the World Health Organization. 16 years after it was put into widespread use, resistance erupted in multiple parts of the world. That resistance spread, and as a result, chloroquine is an ineffective drug in much of the world. It's no longer a first-line treatment, and it's certainly not used in isolation in many localities. There was a combination of drugs, sulfadoxine and pyrimethamine, that encountered resistance evolution about 6 years after widespread deployment. Same story for mefloquine. Atovaquone, there was resistance even in some of the first clinical trials. And artemisinin, which is the current first-line treatment for malaria, and which was introduced in widespread use only about 15 years ago, resistance was first documented by the scientific community in about 2009. And so what the scientific community is now focusing on is not preventing resistance to artemisinin, but containing resistance to artemisinin, limiting it to the geographic location where it first arose. So evolution on the parasite side is something we constantly have to contend with because drugs impose a very strong evolutionary fitness pressure on malaria parasites. So one question is how does this resistance evolve? It always comes up. It seems to come up easily, and it spreads between malaria populations if we do nothing to control it, if we don't change the way we treat people. So one way we can use genomic data, one way we can use an understanding of evolution, is to try to find the genes that are associated with drug resistance. And there are two kinds of signals we can get out of genome sequencing data from malaria parasites to try to find and implicate the genes that are associated with drug resistance. So one is just diversity. So this is a cartoon of a malaria parasite chromosome. And those little orange lines, those little vertical lines, are meant to indicate places on that chromosome where malaria parasites differ within a population. So if you lined up two chromosomes from two different parasites next to each other, and you check to see where they were the same and where they were different, you would find a distribution of positions where the one strain has an A and the other has a T, or one has a G and one has a C. So you can look for areas of chromosomes that have high diversity or low diversity, and areas that have low diversity tend to be associated with drug resistance for reasons I'll explain in a moment. The other signal has a really fancy name. It's called linkage disequilibrium. It basically just means correlation in allelic state. So if you have these variable positions in the genome, we can say that this orange bar here has either an A or a T nucleotide position, depending on what parasite you sample in the population. And this position has a G or an A, and this position has a C or a T. You can look to see how correlated the allelic state is, how correlated the nucleotide letters are between positions, and use that as a signal to try to pinpoint the genes associated with strong natural selection, likely associated with drug resistance. So you can see between these two positions, the A nucleotide is always associated with a G nucleotide. And same with the T nucleotide. It's always associated with an A at the middle position. But between the middle position and this right position, there's a lot of shuffling. The Gs are not always associated with Ts. They can also be associated with Cs. Okay? So when the deck is shuffled, when the alleles are not correlated to each other, that's the normal background state. When we see a strong state of correlation or a strong state of linkage disequilibrium, that's a potential signal of strong recent natural selection. And the phenomena that causes these signals to be informative is called a selective sweep. And here's how this works. So before you have a strong selective pressure in a population, let's say each of those horizontal bars represents a chromosome, and the colors represent a particular version of that chromosome in that population. Let's say there's a mutation indicated by that red bar that confers resistance to a drug. So after that drug is deployed and applied to that population, there's strong natural selection for any parasite who's harboring that resistance mutation. So what happens is that resistance mutation spreads through the whole parasite population, and it drags with it the whole background chromosome that it's sitting on. And what that means is you lose a lot of diversity. So if we look at diversity here, before that selective sweep, diversity is relatively high and uniform. After the selective sweep, there's a big trough. There's a big decrease in diversity within the parasite population in the neighborhood of that gene conferring resistance. And for linkage disequilibrium, the state of correlation, it starts out low. But then after you drag just one version of a chromosome through a population, or nearly all the way through a population, you see strong association of allelic state. And so as a result, the degree of correlation, or LD, is much higher in the neighborhood of that gene carrying that mutation. So we can use these signals and some other signals to conduct genome-wide scans for natural selection in the malaria parasite genome. And this is work done with Diane Worth and Pardis Sabeti and Dan Hartle and other members of the Broad Harvard Malaria Research Consortium. This is a profile of the strongest signals of natural selection in the falciparum malaria genome. And the higher these little piles of balls go on the y-axis here, that means the stronger the evidence that there was recent natural selection in that neighborhood of the genome. And these different colors here along the x-axis, along the horizontal axis, correspond to different chromosomes. So this little orange set here represents chromosome 1, chromosome 2, chromosome 3, chromosome 4. So you can scan across all the chromosomes, having them lined up like this, and look for those neighborhoods in the genome that seem to have the strongest evidence of recent natural selection. And what we see are that some of the strongest signals we find in the genome are sitting right on top of genes that we know through molecular biological work are associated with drug resistance. These are genes that confer resistance to one or more different anti-malarial therapeutics, so the PFMDR1 gene and the PFCRT gene. Some of these signals of natural selection occur on top of genes that we don't understand their function. So we have some more work to do to figure out if these other signals correspond to selection or not. But we know that drugs explain a significant fraction of the strong signals of recent natural selection in the parasite genome. So we can use these signals in cases of evolution of resistance against new drugs to try to find out which genes are actually implicated, which genes are conferring this resistance phenotype in a parasite population. And if we figure this out, we can start to come up with strategies for finding drugs or come up with strategies for combining drugs together that take into account this understanding of evolution that we can build through these types of studies. So here's one approach, one kind of evolutionarily savvy approach that people have begun exploring in malaria as well as in other infectious diseases to counter the evolution of resistance. And this idea is based on the premise that combinations of drugs are inherently harder for a parasite to evolve resistance to than single drugs by themselves. Right? Of course, nobody who has HIV is now treated with a single drug. Everybody gets a multi-drug regimen because HIV will evolve resistance to a single drug in a heartbeat, right? But if you throw multiple drugs at it all at once, it's much harder to evolve resistance. We can take this up a notch and specifically try to find combinations of drugs where the pathogen can just not be simultaneously resistant to all of them at the same time, even if it were allowed the opportunity to try to slowly evolve resistance. So let's imagine a drug target here, symbolized by this kind of orange oval, and two different drugs, A and B. And when this A drug can dock into that drug target, that means it's doing its job and the pathogen is susceptible. So applying that A drug leads to the evolution of resistance to the A drug in the pathogen. And you get a change in that docking site there so that the A drug can no longer stick. So the B job comes in and does its job, the B drug. The pathogen then could try to evolve resistance to the B compound, but evolving resistance to the B compound in this scenario restores susceptibility to the A compound. And so you kind of put the pathogen in an evolutionary trap by creating a system of evolutionary pressures that it can't simultaneously get away from both of them. So this is an approach that Amanda Lukens and colleagues at Harvard School of Public Health have used, in this case trying to pair the drug chloroquine that I earlier mentioned with a new compound, a drug candidate called IDI-3783. That's a catchy name. But it's one of these early stage chemical compounds. So Amanda and colleagues looked at two strains of malaria parasite, the DD2 strain and the 3D7 strain. The DD2 strain is very resistant to chloroquine, but the 3D7 strain is susceptible. And there's a reciprocal phenotype for IDI-3783. DD2 exhibits low resistance to this drug, but 3D7 exhibits high resistance. The difference between these two parasite strains, because we know that the resistance gene for chloroquine is this PFCRT gene here, this transmembrane protein. We know that the difference between 3D7 and the DD2 parasites is that there's a mutation here. There's a lysine that's been changed into a threonine at amino acid position 76. And so DD2 has the threonine and 3D7 has the lysine. So if we take DD2, which is resistant to chloroquine, and we put it under selection in the laboratory, so we expose it to small amounts of IDI-3783 to cause it to become resistant to IDI-3783, what happens is we see a mutation at amino acid position 352. Okay, so a glutamate changes into an arginine. And what happens when you incur this mutation on a DD2 malaria strain is that the resistance phenotype flips. So those DD2 parasites are now not resistant to chloroquine, and they're highly resistant to IDI-3783. So this is an example of a combination of drugs where these two mutations kind of cancel each other out, and where, in theory, malaria parasites would not be able to evolve a successful evolutionary response to this play. So I don't know that IDI 3783 necessarily has a bright future. It's very hard to become a successful drug. There's a lot of attributes that chemical compounds have to have. But this general principle is being used in malaria and in other systems. And it leverages our understanding of how resistance develops and how evolution naturally occurs. All right, so I talked about drugs. But drugs have not been the only evolutionary pressure on malaria parasite populations. Our immune systems have been a really potent evolutionary pressure on malaria parasites for far longer than our drugs have been, because we only figured out how to use drugs in the last 100 years or so. So this is a cartoon of the MHC molecule, the major histocompatibility complex, which in purple, which is expressed on the surface of cells in our body, and which binds to little fragments of the proteins of pathogens and helps elicit an immune response against those pathogens. So what happens, this is kind of the molecular front of our immune system's interaction with a pathogen. What happens is that there's this evolutionary pressure from this binding interaction between our purple MHC molecule and the green protein of a pathogen. There's an evolutionary pressure on the pathogen to incur a mutation that will interrupt that binding relationship and make it not recognized by our immune systems. That creates reciprocal pressure on the human host population to develop a different version of the MHC molecule that restores that stickiness, that binding, that lets us recognize that there's a bad pathogen inside our bodies. And so if you let this process go, it generates all kinds of diversity. It generates a large store of diversity in a pathogen population. It also generates lots of diversity at the MHC loci. And these are one of the most polymorphic variable genes in the human genome. So whether it's mediated by MHC or whether it's mediated by antibodies, another important component of our adaptive immune response, our immune system effectively imposes a diversifying selective pressure on malaria parasite populations. Instead of just driving them to all become resistant to the same drug in the same way, our immune systems cause malaria parasites and other pathogens to be as different and variable as possible so they can try to escape an immune recognition response. So if we sequence lots of malaria parasite genomes and we compare them to each other and we look for the regions of the genome, the genes specifically, that are the most variable in parasite populations, it's a reasonable assumption that that variability is maintained in those genes by the pressure of our immune systems. And so this is an analysis conducted by a collaborator, Alfred Mbabwa from the Gambia. He looked at a good collection of sequenced malaria parasites from the Gambia and from Senegal and West Africa, and made a chart of how diverse these different gene families were in terms of their variability within this malaria parasite population. And so the higher these bars are on this vertical axis, it means the more variable that protein family is. And all of the gene families with variability for this test statistic above 0, where higher values mean balancing selection, the kind of selection that our immune system imposes. All of these gene families that have values above 0 happen to be surface-expressed proteins. They're proteins that the malaria parasite either has on its surface or they get pushed out to the surface of an infected red blood cell. They're precisely the proteins that we would expect would be under the most immune pressure. So we can, I think, take a little bit of pride in knowing that our immune systems, collectively as a species, have created this pattern of diversity in malaria parasite genomes, which is evidence of their attempted escape from our immune systems. And this is practical information because we can use this evidence of immunogenicity, what proteins in the malaria parasite proteome our immune systems most respond to, when we try to think about how to come up with effective vaccines. So malaria is a disease for which there is no licensed vaccine. Malaria is a pathogen that has about 5,000 genes. It's a lot more complicated than polio or most viruses that have a much smaller complement of genes. With all of these complicated proteins, many of which are harboring extensive variability in their populations, it's very hard to create a vaccine that gives significant protection against all the different flavors of malaria parasites that a person might encounter. So we can try to use our understanding of variability in malaria parasite populations to study vaccine candidates and pick proteins to target vaccine towards that are more likely to be effective in this challenging environment. So one thing that we're always worried about when coming up with vaccines is strain-specific immunity. And so here's a little cartoon to show. If we vaccinate someone against red parasites, and a mosquito bites them and they're injected with, let's say, a whole collection of different parasite strains, red, green, and blue. If they're vaccinated just against the red parasites, what could happen is that the red parasites are stopped by the vaccine-elicited immune response, but the blue and green parasites get through, infect our livers, and then infect red blood cells. And that wouldn't be a particularly effective vaccine. So what we can do is we can try to find proteins that either exhibit low amounts of diversity in parasite populations. And if we do have vaccines that target highly variable proteins, we can try to understand what that diversity profile really is in places where we want to apply the vaccine, and perhaps tailor the vaccine even to the local genetic profile of those malaria parasites. So what this is a histogram showing levels of diversity, levels of variability of different malaria parasite proteins across the proteome. So this vertical axis represents the number of parasite proteins in each category, and this represents how high that diversity is, with low diversity down on this end and high diversity on this end. So most proteins are not variable at all in malaria parasite populations. There's a very narrow tail of proteins, though, that extend way out here, and I even truncated this distribution, that exhibit extremely high variability within populations. And for better or for worse, the circumsporozoite protein, which is the target of what's likely to be the first licensed malaria vaccine, the RTSS malaria vaccine, which could be licensed this year, is one of the more diverse proteins in the malaria parasite proteome. So what we can do is conduct genomic studies to try to understand whether the diversity in that protein does confer the kind of allele-specific protection I previously referred to, and we can think about how to boost the efficacy of this vaccine in that case. We can also just look for variability in among parasite populations in terms of vaccine strain matching. So the RTSS vaccine is based on a single strain of the malaria parasite. And the version of the circumsporozoite protein that vaccine uses is at different frequencies in different geographic locations. So here, it's actually the most abundant version of the parasite that we find. The vaccine strain is the most common, but it still only explains about 20% of all the parasites in that population. In other places, the vaccine strain is extremely rare, as indicated by those very thin red bars. So we can use genomics to try to find good targets for vaccines and also try to understand in which settings those vaccines might be most effective by profiling the variability of those vaccine targets in those populations. So that's drugs and vaccines. I'm going to talk briefly about vectors, because mosquitoes are an important component of this equation. You don't get malaria from people without a mosquito intermediary, but it's only Anopheles mosquitoes that are transmitters of the disease. Nobody knows why it's only Anopheles mosquitoes and not Culex mosquitoes or Aedes mosquitoes or other genera of mosquitoes, but Anopheles mosquitoes kind of have this market cornered. This trait is a really complicated trait because it's driven by a lot of different features that make Anopheles the just right genus, and some species of Anopheles the just right species to spread malaria. There are about 400 species of Anopheles, but there's only probably about 30 to 40 that are important vectors of malaria. So there's certain mosquito species within this genus that have the right combination of chemoreception, what they can smell, salivary proteins, insecticide resistance traits, and immunity, the way that they deal with their own immune systems with the malaria parasite that makes them effective vectors. So one project that we conducted at the Broad, along with collaborators from Notre Dame and from all around the world, was to sequence a collection of 16 different species of Anopheles mosquitoes. Some of them are consummate vectors, like Anopheles gambiae, and some of them are not great vectors at all, like a close relative of Anopheles gambiae. Here's Anopheles gambiae at the top of this evolutionary tree. Anopheles quadriannulatus right here, it's a morphologically indistinguishable. You couldn't tell these two apart by eye if your life depended on it. Anopheles quadriannulatus is a terrible vector because it prefers to bite cows instead of humans. So it doesn't transmit human malaria. So we sequenced all of these species so we could compare their genomes and find the genes that are related to this constellation of traits that make certain species good vectors, good spreaders of malaria. So we aligned these genomes, we studied the protein sequences, and we came up with measures as to how quickly these proteins were evolving in this evolutionary tree. And then we ranked proteins. And the very fastest evolving proteins in mosquito genomes happen to be gustatory receptors and olfactory receptors, little proteins in the antenna and various other parts of the mosquito anatomy that do all kinds of things. They help them find mates, they help them find resting sites, but they also help them find hosts. So we think that this protein family may be important for recognizing humans as a choice meal. Another category I want to call out are salivary proteins. These are mosquito proteins that are injected when the mosquito bites you. The mosquito doesn't just suck blood up, it injects saliva with anticoagulant properties and a whole other cocktail of proteins that help with the feeding biology. So these are evolving quickly. And then finally, immunity. So immune modulation. Mosquitoes are infected by all kinds of things, just like us. They're infected by viruses, they're infected by bacteria. They are infected by malaria parasites. They don't like to be called vectors. I think they would prefer to be called other hosts of malaria. It's not a willing role that they participate in. So we can take a closer look, and we can look at the evolutionary rate. This is two measures of evolutionary rate, one on the x-axis and one on the y-axis. And basically what we can do is we can look at all the genes. Each one of these gray dots is a gene in the mosquito genome. And the evolutionary rate is measured across all the genomes we sequenced. We can look for those genes that exhibit the highest level by both of these measures up in the upper right-hand corner. And these two gene families in the mosquito genome, leucine-rich repeat immune proteins and thioester-containing proteins, both happen to be proteins families that contain members that we know are critical for transmitting malaria. Some mosquitoes have an immune response that quashes any malaria parasite that comes in with a blood meal, but other mosquitoes allow the parasite to develop, and that makes them good at transmitting it to the next human. So we can help to zero in using this comparative evolutionary approach on those proteins in the mosquito immune system that are important for this biology. So we know these genes. Is this just of academic interest, or what can we do with this? What's the practical application of this kind of knowledge of these genes that affect how they smell hosts and how they respond to their own immune challenges? Well, one avenue, one exciting new avenue, is to kind of rewrite the rules of evolution and to use a system called a genetic drive system, where we can try to push genes through a population even if they're not selected by evolution, even if they don't confer a fitness advantage. And we can do that using this CRISPR tool, which many of you may have heard of. If you haven't heard of it, you probably will hear of it sometime. It's a nuclease that lets you very specifically alter a genome. And if you couple this alteration tool with another genetic trick called a drive system that will push it through a population and cause it to convert all the members of that population, you can effectively modify a wild population in any way you wish in a very specific manner. So the way this works is that usually if you have let's say you have one mosquito in blue that carries this modified gene and its drive system, and it mates with a wild type mosquito in gray. Ordinarily, when two individuals mate and they differ at a locus, there's a 50-50 chance that the offspring will inherit the modified version of that allele. But with a drive system, all of the offspring always inherit this system, and they always transmit it to their offspring. And so in a genetic drive system, particularly one that's facilitated in a very specific way via a CRISPR endonuclease, what you can do is release some amount of modified mosquitoes in this case, or any organism this applies to, and have that spread through the entire population. So you've effectively modified a population. And so there was a paper that was put out by Kevin Esvelt and colleagues at Harvard in the last year asking the question of can we start debate about this? When is it justified to modify or deliberately eliminate a wild population of a species? This is something where, with mosquitoes, for the purpose of disease control, we can imagine restoring susceptibility to insecticides where resistance has already evolved. We can imagine changing a sensory receptor gene that is involved in a human-seeking host preference. We can imagine tweaking a gene that causes all of the progeny of a mating event to be male. And what that does is it causes the mosquito population to get very small. So mosquitoes have their advocates, and I'm not taking a particular stance on this, but this is the kind of issue that I think as a community, as a species, we are going to have this in our toolkit of possible ways to address infectious diseases and vectors of infectious disease. So I just want to end on an optimistic note. So despite all of those ominous statistics I cited at the beginning of this talk, mortality is actually 47% lower than it was 15 years ago. As I mentioned earlier, the first malaria vaccine to be licensed is likely going to be licensed this year, and we have others following behind it. We have new ways of developing drugs. We don't just have to randomly try different compounds. We don't have to find bark from a tree that was used to treat an unrelated ailment and turn that into a wonder drug. We have smart ways to find drugs, and we have genomic tools to help us watch resistance as it develops, and we can try to stamp it out or at least prevent its spread. And then finally, we have a strong history of local elimination of disease. So even though malaria is still extremely endemic in many parts of the world, it's receding into the past in places like the United States, where we had malaria until the 1940s. There was malaria in the Connecticut River Valley in Massachusetts until the 1940s, when mosquito spraying finally wiped out the short seasonal transmission patterns. So with these tools and with the backing of organizations like the Bill and Melinda Gates Foundation, I think there's a lot of progress to be made in the next few decades, and I think we can really turn the tide against this disease. So that's all I've got, and thank you for your attention.
Why does the Anopheles mosquito prefer to bite human beings?
That's a great question, and I should say that it's only some Anopheles mosquito species that do prefer to bite humans. So we can't attribute all of that biology, all of that behavior, just to chemoreception, because we've compared the genes that we find in Anopheles gambiae, one of the most significant vectors, to other very closely related Anopheles species. And we can't pinpoint one single difference. So it's probably a combination of factors. It's the willingness to breed in small pools of water, such as accumulate around human habitation. It's the willingness to rest and live inside human habitations. All of these factors contribute to, I think, the higher efficiency of transmission by some species than others. But we don't have a single answer for that question yet.
Okay, so the cocktail approach works because if you introduce X, it develops immunity to X, but then if you introduce Y at the same time, it bounces back and forth between those two immunities and can't settle on one. How is that different from simply changing drugs over a longer period of time, revisiting some of the ones that were originally developed a resistance, and like saying going back to chloroquine, after a period of time?
Yeah, so
why does one work versus not work versus the other?
Right, there's a difference in cycle length, and actually people have proposed that in some parts of Africa, chloroquine susceptibility has returned. After we retired chloroquine, at least as a monotherapy, as a single drug treatment, chloroquine resistance actually confers a negative fitness consequences on malaria parasites when you don't have chloroquine drug being distributed to everybody who's sick. So malaria parasite populations have responded by relaxing and going back to their wild-type susceptible form. And so it's been proposed that we can perhaps bring some of these older drugs back into use in localities where susceptibility has returned. It hasn't always worked that way. In some parts of the world, chloroquine resistance has stayed around even when we've retracted chloroquine. But chloroquine certainly finds use in combination therapies in lots of settings. Yeah.
So in the same way that that the mosquito, or that the drugs can provide resistance to malaria, why don't the mosquito, or the parasites adapt to the sickle cell shape of
That's a good question. They may be trying to do so. There's interesting examples. I mentioned the dark gene and how that was an evolutionary response that was apparently largely successful in preventing vivax malaria infection in sub-Saharan Africa. There are certain parts of the world, certain parts of Africa, particularly Madagascar, parts of Ethiopia, where there is vivax infection. And one interesting observation that people studying the genomes of vivax malaria parasites have recently made is that they've duplicated the parasite protein that docks with the dark gene. And so parasites may be trying to compensate for some of these human evolutionary responses to disease. But one factor is that because sickle cell has such a deleterious consequence, it never gets to above a certain frequency in human populations. It gets to a frequency where homozygotes, where individuals with 2 copies of the sickle allele start to become more common, and that impedes the further increase in the frequency of that resistance mechanism. So parasites are always going to find some non-sickle individuals to infect, and so they don't, in a sense, have to evolve around that resistance mechanism.
What actually happens in the red cell, the sickle red cell, that makes it resistant to hosting?
That's a great question, and so in an individual that has 1 sickle copy of hemoglobin and 1 wild type copy, the cells have a more normal phenotype. And exactly how resistance is conferred by this heterozygous state of having 1 sickle version and 1 wild type version is not thoroughly understood. It's thought that perhaps having a little bit of an altered shape could allow the spleen, for example, to more effectively clear infected red blood cells from circulation. But I don't think that's been completely confirmed. We're still figuring out the biology of even these resistance mechanisms that we've associated with malaria for 50 years.
So, Dan, when you looked at the most frequently, the most variable genes in Anopheles, and you thought they were the gustatory and olfactory, have you actually seen an instance of host switching among the infection, or are humans considered the optimal host, or how does that?
So I think the best example is in this Anopheles gambiae complex. So Anopheles gambiae, this very important vector in Africa, as I mentioned, has a sister species that's completely identical called Anopheles quadranulatus. And this is the species that prefers to bite cows or ungulates over humans. So this is extremely recent. These species only diverged very recently. There are not very many differences between their genomes. There's enough genomes that we can't find the one difference that is caused with this difference in host preference. I don't think there's anything intrinsically about humans that makes us a better host than a cow. I think there's plenty of cows and there are plenty of humans in Africa. So in terms of just general abundance, we're both good targets for mosquitoes to bite. But there are people who are conducting experiments where they will make a genetic cross in the laboratory between Anopheles gambiae mosquitoes and Anopheles quadranulatus mosquitoes. And they set up these fancy contraptions where they put the scent of a human on one side and the scent of a cow on the other side. And they put the mosquitoes in the middle and they say, you pick, which way do you want to go? And so when they take the hybrid progeny of these interspecific crosses, they can see what parts of the Anopheles gambiae genome they have in for certain genes and which genes have the Anopheles quadranulatus inheritance, and then try to correlate that with the behavior with biting humans or with biting cows. So we know that the variability in these chemosensation and gustatory receptor genes is probably important for a lot of mosquito biology, but we still need to go back to the lab and do these kinds of experiments to implicate specific genes associated with these very specific behaviors that seem to have evolved pretty quickly. Humans are a pretty new species on the evolutionary scene. Our numbers were not significant in Africa or anywhere else until pretty recently in an evolutionary sense. So it's probably fairly recent that Anopheles gambiae has had such a strong association with humans as a host. We've become a very attractive target.
I'm not trying to send you back to the pufferfish, but I would love it if you might speculate about the third word on the screen up there, about when malaria might be eliminated.
Eliminated. So I should explain these terms because they have a very precise meaning in an epidemiological context. Eliminate means to kind of locally extirpate something, and eradicate is what we did with smallpox and perhaps soon with polio. So eradication basically means extinction in the wild. And I think it's very hard to say. I know that in a lot of malaria endemic countries, the national control programs set very aggressive dates for elimination, goals, because it's very hard to be motivated about something that you say is going to happen in 30 years, right? If we're going to eradicate or eliminate malaria in 30 years, then I'll just finish my sandwich and get around to it later. So I think you see a lot of dates out there, but I think it's reasonable to think that on a time frame of a few decades that we could eliminate malaria from a lot of places. And getting those last hard-to-reach places is not just a biology problem, it's going to be a sociopolitical, economic problem as well. So I think we can achieve huge progress, but as far as getting to that last goal of eradication, that's going to take a concerted effort and probably a few good decades of work.
So Dan, we have a question that was tweeted from our audience for you. What's the current prospect for using bacterial endosymbionts, such as Wolbachia, to control mosquito vectors?
Oh, that's a great question. So there's a great, I talked about genetic modification to try to end on a provocative note. But there's another way that you can modify wild populations, and that's to introduce an endosymbiotic bacterium. So Wolbachia is a genus of bacteria that's an intracellular endosymbiont, and it's found in some shocking fraction of arthropod and even non-arthropod species worldwide. And Wolbachia can have interesting effects. It turns out when you have a bacterium in many or all of the cells of your body, arthropods respond. Their immune response to other infectious challenges is altered. And there's recently been very promising success in using Wolbachia-altered Aedes mosquitoes to control dengue transmission in different parts of the world. So Wolbachia has always been a challenge for Anopheles because until very recently, no one had identified Wolbachia that thrived in Anopheles mosquitoes. So Aedes mosquitoes have Wolbachia. Drosophila fruit flies, which are close evolutionary relatives to mosquitoes, have Wolbachia. But Anopheles mosquitoes had not been observed to Wolbachia. So Flaminia catarrhicea, a collaborator at Harvard School of Public Health, her team recently found the first Wolbachia in wild Anopheles populations. And there's the hope that we can learn from this Wolbachia that naturally infects Anopheles and train up or modify or select a strain of Wolbachia that we could use to seed and infect wild Anopheles populations to render them less effective vectors of malaria. So it's an approach that's akin to genetic modification, but you might argue it's slightly less permanent. Yeah.
Dan, I was blown away by your presentation, and it makes me feel very privileged to be a human in this stage of evolution, where we can actually contain, the wild, not in terms of eradication, in terms of understanding sort of how it fights back against us in our own natural evolution. It's just mind-blowing, so thank you very, very much for this. My question is very geeky in a way that you basically have these very rapidly evolving proteins. And I don't know if you mentioned this already, but could you look within these rapidly evolving proteins for slowly evolving portions, which might be harder to sort of escape for, the natural biology of the virus, and basically use that to, not virus, but use that to then construct, vaccines or something against those specifically, because then you're sort of eliminating the place where natural variation can arise, but then focusing on a small place. And my question is very deep. It's like, is it that our immune system does not see these parts because somehow when the protein gets chopped up, that part does not make it to the immune system? Is it that they're sort of not exposed in some way, or is it that these could actually be good targets?
So that's
I want to kind of rephrase that.
sure. So the question is could we look for non-variable regions of proteins? I think that might be immunogenic, that might stimulate a strong immune response, but which are not variable in parasite populations, perhaps because they're functionally constrained, or perhaps because they've just never been presented to our immune systems in a way to target an immune response to that part of a protein. So perhaps the parasites could evolve around a vaccine targeting these regions of proteins, these subregions, but we wouldn't at least be starting out facing a population that was already diverse in those vaccine target regions. And that's absolutely a possibility. So this RTSS vaccine, that's the subject of a recent subject of a phase 3 trial that we studied at the Broad and that, as I mentioned, is likely to be licensed this year. It really only targets a small region of the circumsporozoite protein. And it's really only about half that region, I would say, that's extremely variable. There's many parts of that vaccine target that are not variable. There's many parts of the circumsporozoite protein outside of the vaccine construct region, outside of the region of that protein that's used to incorporate into the vaccine, that are not variable whatsoever, and which could be immunogenic. And I would say that developing vaccines is still enough of a black art. That natural immunity isn't always a good predictor of being able to provoke an artificial immune response that could be quite effective at controlling disease. So we don't necessarily have to limit ourselves to those variable proteins that have, in the past, engendered a strong immune response and an immune escape response in the parasite. We can look for non-variable proteins that have other attributes that might make them good vaccines, and we can leverage all this other information we have together to try to come up with candidates.
I have a slightly related question, which is, if you have now individuals for whom a vaccine works and individuals for whom a vaccine doesn't work, you can just hypothesize that their immune system has sort of that particular variability. Have you guys done that? Have you then gone back to sequence individuals, basically look at their immune repertoire to basically see if it matches the specific variation of the vaccine that they were subjected to?
Yeah, so this is another good question. Not everyone responds to a vaccine in the same way, and that's in part because our immune systems are very different. So this is a question that's also being asked of this same phase 3 vaccine trial for the RTSS circumsporozoite vaccine. There's a study that we hope to collaborate on to at least start looking at transcriptional profiles and understanding which immune genes are turned on and associated with an effective response and protection against disease, and what transcriptional profiles, what genes are turned on and off and associated with a lack of protection from disease. So there's certainly good work we could do on the host side as well. And this is the first vaccine trial, I would say, that's large enough to perhaps give us good statistical power to try to implicate certain genes associated with good response.